of Slovaks from the paper. Recent variability study in Czechs, the neighbors of Slovaks, has shown that they are genetically similar with adjacent European populations, but characterized by a small frequency of East Eurasian, to 0.8%, and Roma-specific, to 0.8%, lineages, Meliarch et al. 2006b. Therefore, the aim of the present study was to characterize the variation in Slovaks from western and eastern areas of Slovakia, based on variation of the HVSI and HVSII sequences, followed by a hierarchical survey of haplogroup-specific restriction fragments length polymorphism, RFLP, markers. In the above passage they are referring to this paper. More on the haplogroup M in Slovaks. However, in contrast to the previously studied Czech population from Western Bohemia, Meliarch et al. 2006b, samples from Slovakia do not display any East Eurasian NAS. One of the Slovak M haplotype belongs to subhaplogroup M1b and is identical to M1b1a haplotypes revealed in Italians and Bedouins from southern Israel, Oliveri et al. 2006, as well as in Saudi Arabs, Abu Amiro et al. 2007. A second M lineage detected in Slovaks is defined by variants at positions 16129162321623163016304163444. This lineage is identical to those revealed previously in gene pools of the Bulgarian Roma at frequency of 3.6%. Gresh et al. 2001. Based on the presence of the 16,129 variant, Gresh et al. 2001, suggested that this lineage belongs to Indian-specific haplogroup M5. Nevertheless, to determine its exact phylogenetic status we completely sequenced our Slovak sample, SLV 227, and compared it with Indian M haplotypes published by Sun et al. 2006, Figure 1. As a result, we have found that our sample belongs to haplogroup M35 due to mutations at positions 199 and 12,561. Moreover, it shared transition at 15,928 with the South Indian sample T17, from Andhra Pradesh, that allowed us to define a new Indian/Roma branch called as M35b, and on a Roma-related J1 lineage. Previously, we have found that the Polish Roma population is characterized by high incidence, 18.8%, of haplogroup J1 lineage, defined by HVSI motif 1606916126216145162235162621 to 16,271, Meliarch et al. 2006a. This and a similar haplotype, lacking only the 16,271 transition, are very rare in European Roma populations, being found only in the Spanish, Bulgarian and Hungarian Roma, Gresh et al. 2001, Ejid et al. 2007. Among Europeans, such haplotypes have been revealed only in French, 0.5%, Gibbet et al. 2004, Hungarian, 0.5%, Ejid et al. 2007, and Czech, about 3%, Venesic et al. 2004, Meliarch et al. 2006b, populations. In the present study, we have found that 2.9% of individuals from eastern Slovakia are characterized by exactly the same J1 haplotype. On differences within Slovakia. The MDS analysis performed on the basis of pairwise FSD values revealed that Slovak populations do not cluster together. Western Slovaks are located together with the Czechs and Austrians, in accordance with their geographic proximity, whereas Eastern Slovaks are placed close to Slovakians. Figure 3. Anhum Genet, online early articles. Oi:10.1111/J.1469 to 1809.2007.00410.x. Mitochondrial DNA variability in Slovaks, with application to the Roma origin annals of human genetics. A. Maliarchk, M. A. Perkova, M. V. Durenko, T. Venesic, J. Lazar, P. Gamulkak. To gain insight into the mitochondrial gene pool diversity of European populations, we studied mitochondrial DNA variability in 207 subjects from western and eastern areas of Slovakia. Sequencing of two hypervariable segments, HVSI and HVSII, in combination with screening of coding region haplogroup-specific RFLP markers, revealed that the majority of Slovak mTNs belong to the common West Eurasian mitochondrial haplogroups, HV, J, T, U, N1, W, and X. However, a few sub-Saharan African, L to A, mTNs were detected in a population from eastern part of Slovakia. In addition, about 3% of mTNs from eastern Slovakia encompass Roma-specific lineages. By means of complete mTNs sequencing, we demonstrate here that the Roma-specific M lineages observed in gene pools of different Slavonic populations, Slovaks, Poles and Russians, belong to Indian-specific haplogroups M5A1 and M35. Moreover, we show that haplogroup J lineages found in gene pools of the Roma and some Slavonic populations, Czechs and Slovaks, belong to new subhaplogroup J1A, which is defined by coding region mutation at position 8460.